A very good evening, everyone. I hope I'm audible to all. So, myself, Dr. Himani, and I welcome you all in this training program on the behalf of Vital Biotech Agriculture Forum for Technical Education of Farming Society Kota, Faculty of Veterinary Science and Animal Husbandry Ranchi, and Birsa Agriculture University Ranchi. I cordially welcome you all in this 7 days training, international training come workshop on cutting edge innovation in veterinary sciences for a sustainable future. So first of all, I would like to tell all the participants a little bit about the organizers. So organizers are like Vital Biotech. It is India's leading publication, research and training institute. Its goal is to tackle the world's most pressing problem which are related to food and agriculture. Whereas Agriculture Forum for Technical Education of Farming Society Kota, it serves as a spiral between the network of rural communities in India and other stakeholders, such as government, development organizations, and corporate entities. And now about the Birsa Agriculture University, it is a well-renowned name. So this university was established on 26 June 1981, and its inauguration was done by then Prime Minister of India, late Srimati Indira Gandhi ji. So the primary objective of this university is to develop area-specific technology and manpower in the field of agriculture, animal husbandry, and other and forestry for the agricultural development of Jharkhand and economic upliftment of the tribal and other backward classes of the region. The program and activities of teaching and research extension are being carried out through 7 faculties and 12 colleges in this university. Beside this, one college is of biotechnology, one center for agribusiness management, 6 directorates, 3 zonal research stations, farms, one university farm, 16 KVKs, computer centers, central library, ATISC, Radio Haryali, Krishi, Patri, and other infrastructure service units are there in the Birsa Agriculture University. And the College of Veterinary Science and Animal Husbandry was established on November 30, 1961, with the late Colonel Dr. S.M. Ishaq as its first principal. Initially, it was a part of Ranchi University, and then it became a constituent of Rajendra Agriculture University in 1961. It focused on undergraduate programs. Postgraduate programs were added in 1972, and further expansion in 1977. In 1981, this was incorporated into Birsa Agriculture University itself, and since then it is offering UG, PG, PhD, diploma, and certificate courses to various students. This college is also involved in research projects related to animal health and nutrition and conduct extension activity like livestock health camps and farmers education. So once again, I want to congratulate the entire organizing team for organizing this insightful training program, which is specifically for the veterinary sciences students and academicians. So today we are highly privileged because we have so many dignitaries on board. For inaugural session, first of all, I would like to invite Dr. Dinesh Kumar. He is assistant professor cum junior scientist in Department of Animal Nutrition, College of Veterinary Science and Animal Husbandry, Birsa Agriculture University, Ranchi. He is also the organizing secretary of this training program. So before inviting sir, I would like to tell a little bit about him. So sir has earned his BSc in Veterinary Sciences and Animal Husbandry from Anand Agriculture University, Gujarat, and Masters in Veterinary Sciences and Animal Nutrition from the National Dairy Research Institute, Karnal. Sir has completed PhD in Animal Nutrition from Indian Veterinary Research Institute, and since February 2017, he worked as an assistant professor in College of Agriculture at Jawaharlal Nehru Krishi Vishwavidyalaya, Jabalpur. And after that, in September 2022, he began working as an assistant professor cum junior scientist in Department of Animal Nutrition at College of Veterinary Sciences and Animal Husbandry, Birsa Agriculture University, Ranchi. Sir has maintained a stellar academic record throughout his study and received numerous awards, including the National Talent Scholarship, the ICMR Junior Research Fellows, Fellowship, and the Senior Research Fellowship from various funding agencies. Additionally, he is assistant registrar in charge of the college academics and the dairy unit. Sir has published more than 30 research papers in various reputed national and international journals. Also, Sir has written more than 50 articles in various national and international journals. And Sir has extension articles, 15 book chapters, instruction manuals, and a bulletin, which is technical bulletin. Also, Sir has written that. And Sir is a co-PI. He is managing 2 projects with outside funding. He is a lifetime member of various numerous reputed societies and associations like Animal Nutrition Association. And he is regularly participating in conducting various workshops and training sessions. And he is a reputed reviewer of 6 prestigious journals with significant national and international impact. So, sir has so much, so many of the his uh, so many things in his CV. I cannot enlist all of them in such a short span of time. So for now, without wasting much time, I would like to invite Sir on board. So over to you, Sir.
Thank you, Himani. Very good evening to all of you. I am audible?
Yes, sir, you are audible.
Okay. So, very good evening to all of you. So, distinguished guests, esteemed scientists, respected faculty members, dear participants, and fans. It is my great honor and privilege to welcome you all to the 7th Day International Training and Workshop on Cutting Edge Innovation in Veterinary Science for a Sustainable Future. As the organizing secretary, I take great pride in witnessing the convergence of brilliant minds, dedicated professionals, and aspiring scholars who have gathered here to contribute to the advancement of the veterinary science. The theme of this event resonates with the growing need for scientific innovation, sustainable livestock practices, and transformative approaches to veterinary sciences. So today, veterinary sciences stand at the crossroad of innovation, where technology, research, and interdisciplinary collaboration are shaping a new era of animal health, welfare, and productivity. So the veterinary field has always played a crucial role in the ensuring animal health, food security, public health, biodiversity conservation. So, however, with the challenges posed by climate change, emerging infectious diseases, and the growing demands for sustainable livestock production, the need for innovation has never been greater. So, actually, this workshop brings together renowned experts, researchers, academicians, and professionals from across the globe to share their insight, experience, and research findings. So, our objective is to explore cutting edge innovations that will revolutionize animal health care and enhance productivity while ensuring environmental sustainability. So, this training has been meticulously planned to ensure a holistic learning experience for all participants. So, over the next 7 days, we will engage in the interactive lecture, expert panel discussion, and knowledge sharing session covering diverse topics. So mainly we covered the theme area. Just I am introducing the theme area where we discuss in the next 7 days. So mainly we discuss the advances in the assisted reproductive technology and genetic improvement, precision livestock farming and smart technology, emerging infectious disease and one health approach, biotechnology and nanotechnology in veterinary sciences, climate resilient strategy in livestock production along with the mitigation of methane. So this workshop is not just an event. It is an opportunity to collaborate, innovate, and implement solutions that will define the future of veterinary sciences. So I encourage each one of you to actively engage in discussion, exchange knowledge, and explore potential partnership for future research and development. So I extend my heartfelt gratitude to our deans and organizing committee and collaborating institution for their relentless effort in making these events a reality. So I also commend the dedication of our speakers and trainers from various parts of the country who have graciously agreed to share their expertise in this training program. So as we commence this workshop, let us reaffirm our commitment to leveraging scientific advancement for a more sustainable and resilient veterinary sector. So I am confident that the discussion and learning from this event will significantly contribute to shaping policies and practices in the veterinary sciences. So as we embark on this insightful journey, let us collectively work together a future where veterinary sciences continue to be a pillar of sustainable development, ensuring animal health, environmental balance, and food security. With these words, I again welcome you all and wish you an inspiring and enriching experience over the next 7 days. So thank you one and all for joining such a events, for sharing our knowledge to the all the participants and the researcher and scientific community. Thank you one and all, and hand over to the Himani.
Thank you, sir. Thank you for giving the outline of this training program to the participants. And now moving forward, it is a great matter of pride for all of us that today we have the chief guest of today's session, Dr. Sushil Prasad with us. So, sir is Professor cum Chief Scientist and Chairman, Dean, Department of Livestock Production and Management, College of Veterinary Science and Animal Husbandry, Birsa Agriculture University, Ranchi. So, before inviting sir, I would like to tell a little bit about the achievements of Dr. Prasad. So Dr. Prasad is a distinguished academician and administrator at Birsa Agriculture University, Ranchi. Sir has served as Dean of the Faculty of Veterinary Science and Animal Husbandry, where he oversees academics, research, and extension activity. Sir has over 36 years of experience. And in addition to his role as dean, he has held various positions like professor, scientist, chairman, and head of the department, additional director, extension education, additional director, research, cultural coordinator, and has contributed significantly to the university's outreach programs. Under his leadership, the Department of Livestock Production and Management has made notable advancement. And one significant achievement is development of the Banda pig variety, along with development of a suitable dual type variety of chicken named Jassim for rural poultry production under backyard system, which has gained national recognition for its role in improving the socio-economic status of communities, particularly in Jharkhand state. This initiative has been instrumental in enhancing livestock productivity and farmers' livelihood. As PI and co-PI, sir has handled several projects with outside funding, including AICRP on goat and poultry. He holds multiple executive positions in society and is a member of numerous professional society and associations. Sir has published more than 60 research articles in various reputed national and international journals. And additionally, sir has written 30 extension articles, 25 book chapters, and many instructional manuals and technical bulletins. Sir has guided several PhD and PG students in livestock production and management. So the academic journey of Dr. Prasad includes studies at Ranchi Veterinary College and PhD degree from CCS HAU Hisar, reflecting a strong foundation in veterinary sciences. Extensive experience and dedication of Dr. Prasad has positioned him as a key figure in advancing veterinary education and research in the region. So the list which I can see is endless, and enlisting all the achievements in a short span of time is not possible for me. So I would like to invite Sir for his insightful talk with all the participants. Sir, over to you. Sir, kindly unmute. You are on mute. Yeah, now it's fine. Sir, I think... Yes, it's fine, sir. Fine.
Audible I am?
Yes, sir. Now you are audible perfectly.
Okay, thank you. Thank you, Dr. Shivani. I welcome all the participants in this 7 days training program and workshop. So first of all, honorable guests, then our esteemed scientists, distinguished colleagues, as well as our the and dear students and the faculty members, those who are joined this session of training and workshop. I feel privileged and honored that we're standing before you for inaugurating this workshop and training program of 7 days that is on the cutting edge of um, cutting cutting edge innovation in veterinary science for sustainable future. This is a very good topic, and during these 7 days, I hope this will be beneficial for all the participants, those who are attending this training program. Like this training program is going to start from today, that is 20th March to 26th March. So this topic itself indicates that how we are going to make the veterinary science in a sustainable manner. Because the every day we are hearing and there is a global warming, another aspect that is the climatic change. And this climatic change will definitely affect the animal production as well as the animal health. Similarly, the food insecurity. That is the food insecurity when we talk about this, the people, not only the food, but the animal foods are also for the nutritional security. Like the India is a poor country and having the farmers level very much deficient in the nutritional aspect. So we have to think over that also. Then third aspect is that the resource management, deficiency in resource management, because every day the farmers are not getting the profit from the because they are not sustainable livestock production. So the veterinarian should think about the how we are going to make a sustainable livestock production. Because if we are doing a job in veterinary science or animal husbandry side, then this is the very much important thing to think that if a farmer is going to start a business or a income generation venture of animal husbandry or animal side, then he always think about that for the production aspect. And the production aspect, when we go for production aspect, then we have to think that if we, when the animal will how the animal will sustain the production, because in we if we are seeing that the climate change is going on, then definitely it affects the animal health also, and definitely the production aspect also. And that's there are several diseases are emerging due to this climate change. If we go think that last not what last year before, there was a lumpy skin disease in whole Rajasthan. Then there are several mortality in this case of lumpy skin disease, and it has affects the production aspect of the animal. Then we have to think that how we will go for to change the environmental aspect, what type of management is required to make a sustainable animal. Similarly, that if you go for this bird flu, this previously it was only in chicken. I think chicken and duck was the reservoir, but nowadays it happens in all the types of in the farming condition. I'm not saying that in jungle aspect, but it is in the farming condition. This type of diseases are occurring. Then the farmers, those who are doing this either poultry farm or cattle farm or the goat farm, similarly, what happens that the farmers withdraw its business because they think that this is not a sustainable venture for getting earning the money, because the veterinarians have to think several things of this, that is the animal health, food security, public public, even the public health. Veterinarians have to think about the public health also, because the several zoonotic diseases which transmit from animal to human being or human being to animal. So we have to think about the zoonotic diseases, then public health, and then one thing is that biodiversity, biodiversity conservation. Biodiversity conservation, we are hearing every day that this breed is going to extinct. How? We are not conserving our pure native breeds. So the veterinarian should think also for this, that the gir, like the gir cattle, are more in Netherlands and other countries than our country. Only one state is. Similarly, the area-wise specific breeds are available. So we have to go for conservation, like in Jharkhand state. I am saying about the Jharkhand state, there is no cattle breed itself. But if you will see the cattle breed here are of the smaller in size and having a different characteristic feature. So we have to conserve these breeds also. Similarly, I have recognized one breed of pig. It is in the village condition, and we have to conserve that breed also. The NBAJ are having several programs for the veterinary for conservation of breed, and by this conservation we have developed several breed. We have protected it. So innovation regarding, because in our training topic is the innovation. Innovation is not a single thing that in we can do it in isolation. We have to combine with the effort with the scientists or researchers, along with the farmers, and also with the industry people, farmers, and the researchers, and as well as those policy makers, those who are making the policies for animal husbandry or animals. This they should be included while when we are doing any innovation work, because we have to learn also from the farmer what they are doing, what their traditional method. So by traditional method and as well as the recent technology, involve it and go for new innovation. So this is, I think, this topic will definitely benefit for the all the participants, as well as I say that the students, those who are involved in this, they will think for future aspects of this, and the faculty members, those who are those who have joined this training program or involved in arranging this program. So they will definitely be give last we will maybe may come in conclusion that what is the output of this training after in the valedictory session of this training program. Definitely some recommendations, some uh, should come before this, so that that will be produced before the policy maker should go to the policymaker, that this is the aspect of the veterinary science and how we are going to make the animal husbandry or veterinary science as a sustainable venture for the farmers. So at last, I congratulate to all our organizing secretary, Dr. Dinesh. He has made a very good effort from last, I think, 1 month. He is trying to make it successful, and I congratulate all the faculty members for this, and I give the message to the students that they should must join this training program so that they should learn something from the our senior colleagues, senior researchers, and what is the views of the industry regarding to make it sustainable. So at last, my all the good wishes for the training program and this workshop, and I hope that it will be we will have a grand success of this training program. Thank you very much, John.
Thank you, sir. Thank you for your good wishes and about explaining this program to the participants. So moving forward, dear audience, we have a guest of honor of today's session, Dr. M.K. Gupta. Sir is University Professor, cum Chief Scientist and Chairman, Dean PG, HOD in charge Department of Animal Nutrition, College of Veterinary Science and Animal Husbandry, Birsa Agriculture University, Ranchi. Although due to his busy schedule, sir is unable to join this session, but he has conveyed his good wishes for the smooth organization and success of this training program. So from this podium, I wholeheartedly thank Dr. M.K. Gupta, sir, for his blessings. And now I would like to invite the next guest of honor of today's session, Dr. Ajit Kumar Sinha, sir. So, sir is ex-university professor, cum chief scientist and HOD, Department of Animal Nutrition, College of Veterinary Science and Animal Husbandry, Birsa Agriculture University, Ranchi. So, sir is a retired professor, and about talking in the achievement of Dr. Sinha. So, sir has been graduated from this college only. And subsequently, sir has done masters in veterinary sciences from Indian Veterinary Research Institute, Ijatnagar. And then sir has completed his PhD from Birsa Agriculture University, Ranchi. So after completion of his master's, Dr. Sinha served the government of Sikkim as an assistant research officer and later joined as a junior scientist in AICRP on sheep breeding for fine wool from December 1976 to April 1982. Subsequently, sir joined this esteemed college as an assistant professor, advancing to the position of university professor and head, from which he retired in 2012. Throughout his tenure, Dr. Sinha was actively involved in UG and PG education as well as a range of research projects within the department. He guided 12 master's students and published numerous research articles in reputed national and international journals, which has contributed significantly to the field of animal nutrition. So today we are very honored to have Dr. Sinha with us in this virtual training program. So, sir, now without wasting much time, I would like to invite you for your session. So, thank you, sir, and over to you.
Thank you. Good evening to all. As Dr. Dinesh introduced the topic, what happened? Am I visible? No.
Yes, sir, you're visible and you're audible also.
Okay. So, as you just said, that I was there in the animal nutrition department and I retired in 2012. 12 years have passed. 12 years means a jug has passed. This is the 13th year of my retirement. But when I was informed day before yesterday only that there is a training program and workshop you people are organizing, and Dr. Gupta said me to join this, I was very happy. I was very happy. So it gives me very immense pleasure to be with you, the young scientists. So actually, I won't take much time, but what I want to say in our time also and nowadays also, the subject matter specialist, I mean, in veterinary, I will talk about veterinary. In veterinary, there are so many departments: nutrition, medicine, surgery, and so on. So what we do, how we organize a training program or a seminar, we organize it on this subject wise. Only the student going in that subject or the subject matter specialist, they are organizing, they are participating in seminar, symposium, workshop, training, like that. But what I found, that your topic, cutting edge innovation, I found that it is, no doubt, it is beneficial for the scientist, for the student, but it is more beneficial to the to the veterinarians, field veterinarians. Hmm. You see, field veterinarians are also a qualified veterinarian. You people, we have qualified them, we have sent them to the field, but once they go to the field, whatever is there in their mind, whatever is their ability, they serve the people, they serve the farmer, and to their best ability, no doubt. And they retire in that. They, after passing veterinary science, they and joining the government or any organization, they never find the opportunity to know that what new innovations, what new discoveries, new technologies, what is there, they do not know. So by reaching to those veterinarians through this type of training program is very, very important, because once they go, they do not know that what is happening in this subject, what is happening in that subject, what new discovery, what new technology. Yeah, because they are in the field. You must see that here in the department, here in the institute, if I am professor of nutrition, I am professor of nutrition. Yeah, someone is Dr. Sushil. Once he was also my student. So he is now LPM specialist. But the veterinarian in the field, particularly, is, you can say, that jack of all, master of none. He has to perform everything. He has to see the breeding program. He has to see the feeding and management program. He has to do the surgery. Everything. He is the best clinician because he has to treat the animals. So he must know that what new technologies have been evolved, what new development has taken place in a medicine, nutrition, management. These are, I'm naming these subjects because these are the main subjects which they find in the field. For institution, it's okay. And besides these, extension work as per the program of the government, they are undertaking that. So you people have organized and gave the title, this your title is very lucrative from my point of view, and cutting edge innovation. You people will be discussing, your other participants will be discussing about this, and the farmers with the veterinarians, either they have joined or you reach them through some institution like veterinary college or whatever it may be. You tell them that after your passing, when you passed, this was not there, but now it has evolved. Simply they know about what I feel, that simply they know about the medicines, because this various sales manager, what you call marketing persons of that various company, they reach them and tell them, sir, this is good for this disease, that. But in other subjects, say for example, if I am a nutritionist and no nutritionist reaches there in the field and tell them that no, you adopt this thing, theek hai, we have trained you to that you feed like this, you balanced feed should be there, and how much and what to feed to the milk cattle, what to feed the growers, newborn, like that. But there are, apart from these, there are many things which they must know, that when there is shortage, say there is famine or flood or whatever it is there, what should be fed. Particularly in Jharkhand, everywhere byproducts are being used, no doubt. Byproduct of that particular area specific byproducts. In Jharkhand, I have seen that Karanj is in abundance, Kusum is in abundance. So we have tried. So we have AICR. We were having an AICRP on this utilization of agro-industrial byproduct. In that, we have tested and we have recommended that this much current should be fed to this category of animal. This much kusum should be fed. Sal seed is very common in Jharkhand and Odisha, Chhattisgarh. So this type of things should be here. So I mean to say what I mean to say, you have got the point. So you discuss the new innovations, new technologies, and it should not be only with the scientist in the institution or the companies working like in that. You must reach to the veterinarians. They are your representative in the field, because all the time you cannot be in the field. But the veterinarian in anywhere in India, the field veterinarians are there to execute your recommendations. So what I say that if they will know, then they will improve themselves so that they can serve the. So now by embracing this cutting edge innovation, they can improve the animal health and welfare. They can enhance the environmental sustainability. They promote the human health and well-being. Now support sustainable agriculture and food production. This type of approach will ensure a healthier, more sustainable future for the animal, for the human, and I think for the whole planet. So with these words, I close my this, but I wish all the success to this training program and workshop, and I thank you people also for organizing such type of very useful training program. Thank you very much.
Thank you, sir. Thank you so much for enlightening us with your insightful words. And thank you for joining us for today's session. So moving forward, I would like to invite our next guest of honor, Dr. Praveen Kumar. So Dr. Praveen Kumar, sir, is HOD Assistant Professor cum Junior Scientist in Department of Veterinary Medicine, College of Veterinary Sciences and Animal Husbandry, Birsa Agriculture University, Ranchi. Dr. Praveen Kumar sir has graduated from the esteemed College of Veterinary Science and Animal Husbandry located in Ranchi, and he is driven by a passion for advancing his knowledge and expertise. So he pursued a master's in veterinary science from College of Veterinary Science and Animal Husbandry, Birsa Agriculture University. And for deeper insights into this field, Dr. Kumar has earned PhD from College of Veterinary Sciences, G.V. Pant University, Pantnagar. And his professional career he marked by his significant role as a head of Department of Veterinary Medicine, where he demonstrated leadership and innovation. Dr. Kumar's scholarly contributions are noteworthy. He has published over 56 research papers and has garnered recognition in numerous prestigious national and international journals. His exceptional work has led him to receiving several awards and medals throughout his career. Notably, he was honored with the Excellence in Teaching Award in 2021, acknowledging his dedication to education, and he got the Distinguished Scientist Award, reflecting his impactful contributions to veterinary sciences. Sir, we are honored to have you on board today. And now I would like to invite you to enlighten our participants with your knowledge and with your words. So over to you, sir. Sir, you are on mute.
Yeah. Are you
Yes,
audible?
sir.
Oh,
Yes,
good
sir,
evening.
you're audible.
Thanks, Imani. Good evening to all. So thanks for giving the opportunity to speak as a guest of honor for the inaugural session of the 7 days international training cum workshop on the topic Cutting Edge Innovation in Veterinary Science for a Sustainable Future. I congratulate the organizers for selecting such a nice topic for workshop. It is a truly privilege to be a part of this important event. And during this workshop, the presentation and discussion will be truly enlightening, and I am confident that the knowledge shared will be contributed for the advancement in the veterinary science for a sustainable future. I look forward to seeing the impact of this workshop on the field, as earlier told by our Guruji, Dr. Ajit sir, and observe how this workshop affects the profession. Many innovations are being used in the veterinary science to improve animal health and well-being, and one of these is the machine learning and AI algorithm, which can be used for the analysis of data from various sources, including health records, imaging studies, and lab results. This will provide the veterinarians with more comprehensive and detailed information about the animal condition, leading to faster and more accurate diagnosis. This is the one field and innovation which can be discussed during the workshop, and I have seen that this topic has been included by one of the speaker. This AI will assist in the treatment planning by identifying patterns and predicting outcomes, potentials leading to better treatment decisions and improved patient outcome. This can ultimately result in improved patient care and quality of the life. As I am a clinician, so I will mostly talk about this animal health and care. Now, sensors and activity trackers are the another innovations which these devices collect real-time data on vital signs, activity levels, and behavior. And these will allow the veterinarians to monitor animal health and identify potential issues early. And this another innovations that microphone systems, which can detect changes in animal vocalization and can be an indicator of stress, distress, and illness, particularly in the poultry. So data from digital monitoring can be analyzed to build systems that can predict changes in animal health and well-being of the animals, enabling preventive measures and personalized health plan. Another for technology, that is 3D printing, are offering new possibilities in the creation of customized prosthetics, implants, and surgical tools for animals. Veterinary clinics can now produce tailor-made solution on site, improving the quality of patient care and outcomes for surgeries and treatment. This cutting-edge technology is particularly beneficial for the pets requiring complex medical intervention. And this 3D printing also offers cost-effective alternatives to traditional method of manufacturing medical devices. Another is telemedicine platform, and this was get popularized during the pandemic. And this remote monitoring tools can provide access to veterinary care in the remote area or for the pets with the chronic condition that require ongoing monitoring. Veterinarians can conduct consultation and provide advice using video conferencing and other technology, improving access to veterinary services. This shift is not only reduce the stress on pet by avoiding unnecessary travels to the clinics, but also improve the access of the veterinary services for the pet owners living in the remote area. Advancement in the imaging technology, such as digital radiograph, advanced imaging equipments provide veterinarians with clear and more detailed imaging for the accurate diagnosis. Molecular diagnosis is the another one which has been used for the genetic testing and biochemical analysis. And this can help identifying the underlying causes of disease and predict an animal's susceptibility to certain condition. Another is cloud-based practice management system. This innovation is also used in the current scenario, and this will streamlining the workflows within the veterinary clinics. This system enhance client communication, improve the efficacy of the veterinarian services and medical histories. So secure cloud-based platform facilitate the sharing of information between veterinary clinics, specialists, and pet owners, contributing to more cohesive approaches to pet care. Blockchain could completely change how patient data is shared and stored, ensuring secure and unchangeable records. Electronic health record system streamline clinic workflow, improve data security, and support better treatment outcomes. These are the area where the work is going on, and these are the innovations in which the veterinarians are working and giving the better treatment plan for the animals. I am suggesting that data from various clinical veterinary clinics and research institutions can be pooled to create a larger database, enabling researchers to identify new treatment protocols and emerging diseases. This can be done and with the help of the government and the research institute. And I hope that many innovation will come and will help in the treating management of the animals. I once again congratulate organizer for choosing such a good topic for the discussion, and hope that after 7 days there will be some outcome of this workshop. Thanks, organizer. Thanks, Imani.
Thank you, sir. Thank you for your insightful talk with all of us. And now, next I would like to move forward, and I would like to invite Dr. Dilip Kumar Yadav. So, Dr. Dilip Kumar Yadav is an assistant professor cum junior scientist in College of Veterinary Sciences and Animal Husbandry, Birsa Agriculture University, Ranchi. So, before inviting sir, I would like to tell all of the participants a little bit about the achievement of Dr. Dilip Kumar Yadav. So, sir has earned Bachelor of Veterinary Sciences and Animal Husbandry degree from Sardar Kushinagar Dantewada Agriculture University, Gujarat. And after that, sir has done Masters in Veterinary Sciences with the degrees of specialization in veterinary gynecology and obstetrics from Pandit Deen Dayal Upadhyay पशु चिकित्सा विज्ञान विश्वविद्यालय एवं गौ अनुसंधान संस्थान मथुरा, and then sir completed his doctorate in veterinary gynecology and obstetrics at NDRI Karnal. So since June 2023, Dr. Yadav served as an assistant professor at RPS College of Veterinary and Animal Sciences, and in February 2024 he transitioned to the role of assistant professor cum junior scientist at College of Veterinary Sciences and Animal Husbandry, Birsa Agriculture University, Ranchi. So throughout his academic career, Sir has maintained an exemplary record, receiving several awards, including the National Talent Scholarship, Institutional Fellowship, and award for the best master's thesis. And Dr. Yadav has published over 20 research articles in various esteemed national and international journals, focusing on areas such as semen biology, infertility, and animal obstetrics. Additionally, Sir has authored 20 extension articles, 10 book chapters, and 4 instructional manuals. Sir is a life member of various professional societies and associations, including Animal Reproduction Society. So, Dr. Yadav is actively engaged in conducting various workshop and training sessions, which aimed at enhancing the knowledge of both academics and farmers regarding advancements in animal sciences. So achievements of Dr. Yadav are endless, and we are not able to enlist all of them. So today, Sir is joining us for a vote of thanks. And with these words, I would like to invite Sir for his vote of thanks. And I'm handing over the virtual stage to Dr. Yadav. So over to you, Sir. Sir, you are on mute.
I am audible.
Yes, sir, you're perfectly audible.
Thank you, Dr. Himani. A very good evening, everyone. It is a truly a privilege connecting with you all today in 7-day international training and workshop on cutting-edge innovation in veterinary science for a sustainable future, and expressing my heartfelt gratitude to all those who have made this event possible. First and foremost, I would like to extend my sincere thanks to Honorable VC, Birsa Agricultural University, Ranchi, Dr. Chandra Dubey sir, for their unwearying support for conducting this remarkable event. Your commitment to support promoting such meaningful initiative has felt a lasting impact on all of us, and we are grateful for your kind support in organizing and facilitating this event. A very extraordinary mention goes to respected Dean, College of Veterinary Science and Animal Husbandry, BAU Kanke Ranchi, Dr. Sushil Prasad sir, for their invaluable leadership and guidance, your constant encouragement and vision were pivotal in the successful execution of this event. Your support has been the cornerstone of its success. We are truly honored to have had your involvement in these programs. Dr. M.K. Gupta not able to join us because some urgent work. I would like to special thanks to convener Dr. M.K. Gupta sir for their outstanding leadership and guidance throughout the planning and executions of the event since it is beginning. Your visions and relentless effort have played a crucial role in the success of this event. I also extend my sincere thanks to guest of honor Dr. Ajit Kumar sir uh, and Dr. Praveen Kumar sir for your immense support and for working so closely with the team to ensure every aspect as well as taken care of. I would also like to express my appreciations to organizing secretary Dr. Dinesh Kumar sir, Dr. Yasas Pulu sir, whose tireless effort in shaping and managing executing this event at national and international level from its inception. I would like to thank collaborating partner Dr. Jitendra Mehta sir, Biotech, for their visionary directions and technical logistic support throughout the course of this event. I heartfelt thank to all the esteemed faculty members and technical staff of College of Veterinary Science, Animal Husbandry, BAU Kanke, for your dedications and meticulous plannings, which help in making this event a smooth and seamless experience for all participants. I must also thank the organizing committee as whole of your collecting hard work, cooperations and patience. Lastly, I would like to thank honorable speakers, participants, students, anchor, and other who attended today. Your presence and participations made this gathering a memorable one. And we hope you leave with new knowledge, idea, and inspirations. Once again, on the behalf of organizing committee, I personally thank you to each and everyone who play a crucial role in making this event a success. We look forward to future opportunity to come to come together and learn from one another. Thank you, one and all. Thank you.
Thank you, sir. Thank you so much.
I hand over the screen to Dr.
Thank
Himani.
you, sir. Thank you so much. And with this, we are moving forward for today's session. So this evening, we are indeed fortunate to have our respected and honored dignitary, Dr. Manoj Kumar. So before inviting sir, here is a brief introduction about sir. So Dr. Kumar is a professor of veterinary microbiology at Bihar Veterinary College and Bihar Animal Sciences University, Patna. So sir has completed his bachelor's in veterinary sciences and animal husbandry from Birsa Agricultural University, Ranchi, and master's in veterinary sciences and PhD in veterinary microbiology from Dr. Rajendra Prasad Central Agricultural University, Pusa. So Dr. Kumar has made notable contributions to nanotechnology by developing innovative eco-friendly synthesis methods for carbon dots and nanoparticles, optimizing their size, shape, and surface properties to enhance their therapeutic potential. His work has demonstrated the effectiveness of carbon dots in antimicrobial therapy, notably against multi-drug resistant pathogens, while also exploring their use in drug delivery and diagnostics. Additionally, Dr. Kumar has engaged in interdisciplinary research and he has collaborated with IIT Patna to integrate nanotechnology with microbiology and immunology and is currently leading a project focused on developing biomass-derived carbon dot-based bioadhesive for improved antibacterial and wound healing properties. So beside this, sir has many more other achievements, and these all achievements are very amazing and noteworthy, but enlisting all of them is not possible at this point of time. So I humbly request sir to share a few words of encouragement with the participants and deliver his speech. So with this, I would like to hand over this virtual stage to Dr. Manoj Kumar. So over to you, sir.
Thank you, Mani. Am I audible?
Yes, sir, you are audible.
Yeah. First, just with folded hand, I wish to my guru, Dr. Ajit Kumar Sinha, sir. I am being a student of undergraduate. He taught us nutrition. So it's a blessing of, you can say, my teacher. So I'll try. It's an embarrassing moment because from where I got the basic education. So I still remember those days when we were studying at the Ranchi Veterinary College campus. And today I am fortunate enough and can say feeling proud that I have made such a effort that I could speak before my guru who is sitting and ready to listen me. So survey usually rate me that how much the student has progressed. It is up to him, but I'll try my best, sir, just to show you that how much have pampered us and how much we have can say spread all your ideas and knowledge. Okay, sir. Okay, Dr. Dinesh, please, can I share this slide from here also?
Yes, sir. You can share the slide, sir.
Okay.
Yeah, kindly put it on the slideshow mode. Yeah, it's perfectly all right.
Okay. So just have an introduction. Thereafter, we will be switching to succeeding slides. Okay, the topic given to me discuss with is insight into nanotechnology. So just I have been working for 3 years, almost 3 years. I'm also new in this field on nanotechnology because earlier it was an experience of just having theoretical knowledge. So whatever all the people are knowing what is nanotechnology and what are the progresses we are going. So somehow it was an accidental entry in the technology because I've been involved in this microorganism and pathogens like that. So somehow in an encounter, it hit it to my mind that why don't we try for it? Okay, and for that time I was exploring for some other and gradually by
Sir,
working, it came to my mind
Dr.
that
Manoj,
we can also work on this antimicrobial resistance.
Dr.
This
Manoj.
AMR is a very burning issue. So by doing so, I have diverged myself into many areas. So just let me share my experiences.
Yes, sir, yes, sir.
Yeah. So
Dr. Manoj, your
antimicrobial
voice is
resistance,
clearly
it's a very
not audible.
buzzword, okay, among the medical veterinary, basically in medical science, because there are like super bug and all that. Many
Hello,
can say anti
Dr.
microbial
Manoj, your
resistant
voice is not
pathogens are
clear.
emerging. We are also getting some result because see, just like ampicillin are the microbes are resistant to ampicillin. Never get the antibiotic sensitivity, can say satisfactory sensitivity or zone of inhibition with ampicillin. Cefotaxime, these getting can say total resistance. Seldom we get susceptibility towards cefotaxime, ampicillin, like that. Penicillin has totally, you can say, we are getting results that they are resistant because they are not effective against any type of bacteria. So we are experiencing regularly in our laboratory through clinical samples. Okay, so it's a MR is a global impact. Just it is estimated that 7 lakh death occurs due to antimicrobial resistance because some of the infectious pathogens could not be cured or inhibited by the you can say antibiotics. Okay, and it is projected that 10 million deaths will be around 2050 if unchecked. So it is
Hello,
going
sir.
to a big problem. Hello.
Sir, audible. Sir, your voice is not clear actually.
Okay.
So I think due to the
You are getting
earphones, it's not audible. I think it will be clear if you have if you remove the
Mic.
earphones and talk.
okay. I think it is
Yeah, you
इसको
can
इसको
remove
now
the.
नजदीक
it's
रखिए.
audible, na?
Yes.
It was away from
That's why.
Yeah,
That's
yeah.
why.
Now
that's
it's
why it
much
was
clear.
not getting. So I'll try. Okay, thank you. Sorry for your inconvenience. So I'm switching here. So there are several mechanisms. Just I am highlighting important point: enzyme degradation, target mutation, efflux pumps, biofilm formation, and all that. There are a list of mechanisms which by which the antibiotic resistance occurs. Okay, the current approach and challenges. What are the we are using traditional antibiotics since long back. Okay, so it targets essential bacterial processes or can say its target is limited. And one thing, it's a very common and proven also that bacteria develop resistance. One thing I tell you with one example that is chloramphenicol. It has become since we have joined the university and at that time chloramphenicol was totally resistant. But in these 1 years, I have the chloramphenicol use of chloramphenicol was almost can say disused in the clinical practices. So now for 1 year, I am observing that chloramphenicol is very much sensitive streptomycin we used to say. So this is the you can say scenario happens. Can say they are narrow spectrum because they target limited number. Suppose some are targeting gram-positive bacteria, some are targeting gram-negative bacteria. There are toxicity problems also because they causes some side effects also. Can say antibiotics. And the rate at which the new resistant organisms are evolving, so the discovery or development of new antibiotics is not to that pace. So we sometimes face problem because we have to use multiple antibiotics or concept some can say cocktail like therapy by combining antibiotics and all some chemotherapeutic also. Yeah. And difficulty in is the most difficult is that these all drugs are carried, the antibiotics are carried to the different parts of the body through blood circulation. So it is a normal physiology that some of the organs are not getting blood, they are not in touch with the blood circulation directly. They are somehow getting their nutrient or whatever requirement is indirectly. Okay, so for that reason, we can't can say use antibiotics because it is not reachable in that much quantity. What is it is can say
Sir,
required.
Dr.
Okay.
Manoj, sorry to interrupt you, but अगर आप ये earphones शायद निकाल देंगे तो अच्छे से आवाज आएगी.
क्या ईयरफोन का? हाँ सॉरी
सर
मैडम.
निकाल ही दीजिए, प्लग आउट कर दीजिए इनको क्योंकि इनकी वजह से ना वॉइस आपकी प्रॉपरली नहीं आ पा रही है. हेलो. I think It will be okay without earphones. We can try without earphones as well.
ईयरफोन से कुछ मुश्किल हो रहा है।
हाँ,
विदाउट ईयरफोन आ रहा
उसके
है,
बाद
अभी
ज्यादा अच्छी
मैं
आवाज आ
निकालता
रही
हूँ।
है। हाँ, अभी
जस्ट
बहुत अच्छे से आ रही है।
बहुत
हाँ
अच्छे से आ रही है सर, अब ठीक
अभी ठीक है ना?
है जी।
हाँ, अभी बहुत अच्छे से आ रही है।
हाँ हाँ, मुझे बताएंगे ना, मैं सोच रहा हूँ ये मिल रहा है ऑलरेडी।
नहीं नहीं, उससे उससे डिस्टर्ब हो रहा है, उसको निकाल दोगे तो अच्छे से आ रही है।
अच्छा ठीक। Okay,
जी
so
सर।
there are alternate methods of dealing with the pathogens. Mass therapy is very can say recently emerging can say one of the tools in the hands of therapeutic can say people. So fast therapy it is in the on the way. It is still to be validated. Some probiotic prebiotics are also used just to can say nurture this immune system. Immunotherapy is also used because some antibiotic or immunomodulators are given so that the host can fight with the pathogens. Okay, photodynamic therapy. Okay, so just it is done just to generate the ROS, reactive oxygen species. So that ROS causes this can say inhibition of bacteria or killing of bacteria. Okay, antimicrobial peptides are in can say relatively advanced stage. Still, it is not used. It's just for research purpose. It has been used. And one thing that entered recently, the nanotechnology means we are using nanomaterial, just trying, and so in some can say it is in use also. So we are trying these nanomaterials just to cure the diseases or for some diagnostic drug delivery like that. Okay, so nanotechnology is just use of nanomaterials means you are minimizing or breaking the macromolecules or matter to nanoscale means to the consider diameter of nanometer. So it becomes nano technology. That is the technology what I have experienced from my experience. Earlier, I was just having a theoretical background and I was just talking about the bookish knowledge. But what I thought that I will be sharing in the subsequent slides that break the just like you break the big stone to the powder form entirely by regular grinding and making it smaller like that, and lastly you pulverize it and it becomes powder. So when you can say rub with your hands or pinch it with the finger, so it will be simply like powder. You will not experience any kind of crystal. So this is the analogous story of the nanomaterials using macromolecules and is fabricating it to the nanoscale material, and we are using that nanoscale materials for our purpose. So there are multi mechanism of I can say antibacterial activity, and there is the most advantageous thing what I experienced during my research study, still I am doing also, that you can target it. If suppose you want that I should deliver this drug or conjugated with nanomaterial, so you can send it and transfer it by conjugating it with some antibodies like material or some conjugating with the some functional group like then you can send it to the particular location and there can so it is rather more promising for the cancer therapy in respect to what we are experiencing in the antimicrobial resistance. Okay. And innovation in new material strategies are constantly emerging. Innovations are also can say done day by day. So we are also far behind what the research are going globally. Okay. So it's a simply you can say just I have told you that it's a nanomaterial scale material size matters because can say we are converting any macro molecules to the nanoscale. So there is an increase in surface area, so we can bind use those increased surface area for delivery, drug conjugation, and all that. These are it comes in various shape nanomaterials, rods, sheets, tubes, dots like that. Okay. So there are two types of nanomaterials, means organic and inorganic, whatever we are using. And third is hybrids. It is metabolic. It is synthetic one little bit. So organic nanomaterial means that has been derived from organic compounds. Okay. So even from vegetables and onions and all, we can derive these nanomaterials. Okay. We can synthesize. And there are inorganic nanomaterials which are very famous, like gold nanoparticles, silver nanoparticles, zinc nanoparticles, copper nanoparticles, like that. Okay, so inorganic are very famous. Organic is not so much can say catch the attention of scientific fraternity, but it is more useful, organic nanoparticles. Okay, and if we are combining two types of nanomaterials, basically organic and inorganic, and we are artificially synthesizing some nanoparticles, then we call it as hybrid nanoparticles. Okay, so these are all about nanoparticles. The nanoparticles like metal nanoparticles, which are silver, gold, and copper. Basically, they act by generating ROS, disrupt membrane. Okay, some metal oxides are also just I was telling that it is combined with some functional group. Okay, carbon nanomaterials, CNT, graphene, carbon nanoparticles, carbon dots, like that. There are so many kinds of materials we are using. And polymeric nanoparticles means drug encapsulation target. For target delivery, we combine the nanomaterials with drug, and it is ultimately carrying those drugs to the targeted site. And by doing this, we are treating the animals. Just I told you also
So
that
sorry to
eating
interrupt you, sir, but
hello.
your voice is not perfectly audible to the audience.
But why this is happening? Just
Your voice is breaking. I think there is some sort of problem with your internet or like that.
so internet, it is not a problem of internet. Just let me. Hello.
Yes, sir. Yes, sir. You can continue, and if there is any problem, I will
Please
interrupt
tell
you
me. Okay.
again.
Okay.
Yeah. Okay.
I have to finish from inside. So this is a just pragmatic presentation of different mechanism of action of nanomaterials. So it disrupts the cell membrane of bacteria, and even breaks the electron transport chain. Okay, it damages the protein synthesis mechanism. It damages the DNA. Okay, it causes it generates oxidative.
सर थोड़ा लाउडली बोलिए आई थिंक मे बी
एक मिनट सर,
अब
समझ
अच्छी
गया मैं।
अब अच्छी आ
मेरा
रही है जैसे अभी आपने बोला ना
हाँ हाँ,
1 मिनट
जस्ट
1 मिनट
जस्ट
बहुत
जस्ट
अच्छा है
वो ये ना अभी आया।
अब आ
रुको,
रही
मैं
है अब अच्छी आ रही है यस
एक्चुअली ना मैं बहुत स्लो साउंड में लैपटॉप रखता हूँ। ओके, बहुत मिनिमम, मतलब लाइट ये मेरा है, वो
अभी
मुझे
फिर
अभी
दूर
समझ
अभी
गया।
फिर लो हो गई सर लाइन
अभी आ रही है?
नहीं सर अभी
पता नहीं
बीच में अच्छी आ
क्यों,
गई
अभी अच्छी आ रही है।
थोड़ा सा लाउड आपको बोलना पड़ेगा मेरे ख्याल से।
हाँ
अच्छा अच्छा,
हाँ अब अच्छी आ रही है यस
आ रही है ना? वो
बहुत अच्छी
हाँ
बिल्कुल
हाँ, नहीं, ओके सर, थैंक यू। इसको मैं कोशिश करता हूँ
जी सर
कि
इसी तरीके से कर
वो
लीजिए हो जाएगा
हाँ हाँ, क्योंकि
अभी
मेन
ठीक
अभी टॉपिक जो है ना, प्रैक्टिकल जो मेरा एक्सपीरियंस है, दैट आई एम गोइंग टू शेयर, वो इंटरेस्टिंग। ये सब तो थ्योरेटिकल है, बट
जी जी
मैंने
जी
क्या किया और क्या दिखता है, मैं
हाँ
लोगों
हाँ
को शेयर करता हूँ कि आप भी काम करिए, देन इट विल बी
बहुत अच्छी
ऑफ
आवाज आ रही
यूज।
सर इस प्रेजेंट की बहुत
अच्छा
अच्छी
ना,
चलने
सो
दो सर
हाँ, वो डिसरप्शन ऑफ इलेक्ट्रॉन ट्रांसपोर्ट चेन और डिसरप्शन ऑफ ये डैमेजेस। ओके, peptidoglycans damage, it also causes damage of the cell wall, cell membrane. Okay, and some of the nanomaterials also damages the DNA also. Okay, by thereby inhibiting, or you can say killing the bacterial cell. It enzyme activation and this oxidative stress. What I have worked, that this one will be the promising, because I haven't seen by treating nanomaterials. What I have used carbon-based nanomaterials. So I never seen that bacterial cells are disrupted or like that. Honestly, I am saying that any kind of can say disruption or cell damage or cells are not visible because they were clearly visible under the microscope after gum staining. Okay, but the number was low. Whatever I charged with this nanomaterial, the initial concentration of bacteria that I have counted earlier through dilution method, and after treatment, the number of bacteria declined, but there was no disruption. So I can't say surely because this one is theoretical one. I never experienced, but this ROS, reactive oxygen species, I have confirmed it. So these ROS is actually, to my experience, it is can say the main arsenal of nanomaterials to kill bacteria. There are some can say others can say methods also, enzyme activation, ribosomes assembly, like that. So these all are can say mechanism of action. Different type of nanomaterials are acting in their own way, and but somehow it is a summary of all the mechanism that has been proven or available in the literature. Okay. So just in table form, I have just can say, prepared for this audience: cell membrane disruption, reaction of ROS, enzyme inhibition, DNA damage, protein synthesis. And the most important is it inhibits the biofilm formation inhibition, because biofilm formation is one of the most virulent factor with the bacteria, especially this E. coli and Pseudomonas. E. coli is causing a lot of problem. Because in humans, I have been discussing with one of the urologists that this biofilm formation is the root cause of UTI infection in human beings. He was just telling of his experience that this biofilm formation, once the biofilm antibiotic therapy is given, the bacteria gets inhibited, and the host thinks that the infection is over. But after sometimes relapses occurs due to biofilm formation, and nanomaterials could be, we can say, very helpful in disruption of this biofilm, because it is not a easy job just to break the biofilm of the bacterial cell, especially this is Staph aureus, E. coli, Pseudomonas, like that bacteria. Okay. So now what I wish to share my experience. Just I am working because 2, 3 projects are running in parallel. But one, the basic one from where I have started, just I wish to share this antibacterial and wound healing properties of bioadhesive biomass derived from carbon dots. It was a project in collaboration to IIT. Still, it is can say in use, and we are working. Okay. That this optimize this carbon dots derived from biomass to evaluate the antimicrobial properties. That was my kit. And besides this, we have been targeting that we should replace suture with this material because it acts as a bioglue. So let me explain whatever I have experienced, because the carbon nanomaterial, there is a difference. People are often confusing that carbon nanomaterial and carbon dot. So carbon nanomaterial is a nanomaterial, but carbon dots are also nanomaterial. What we have synthesized, you can say it was can say our idea. Just it was a novel one, and it is below 10 nanometer. Then we call it as or can say treat it as carbon dot. Okay, so below 10 quasi-spherical species, because it is not as entirely spherical. It is a quasi-spherical one, and its size is lesser, and it is surrounded by so many species, like say carbonyl, ammonia, hydroxyl, like that. Okay, and it can be synthesized by top-down and bottom-up process. So top-down process is used for this research purposes, but bottom-up means from lower to higher carbonization of a small molecules. These industries people can be used. So CDs are divided into 4 classes: these carbon nanodots, carbon quantum dots, graphene quantum dots, and carbonized proton. There are some can say chemical property differences in the chemical properties. So I am not going in detail. Just I did. So how to synthesize these carbon dots? I am telling that I haven't used, I was just initially planning to use gold nanoparticles. Okay, so it was interesting, appealing for me that people are telling at time gold nanoparticle, gold nanoparticle. I used to hear and think that what these people are doing, what is it is all about, and I could not understand even. It was a totally theoretical one. So I with Mr. Das, I concept planned that why don't we work for gold nanoparticles? So by reviewing the literature, it came to mind that why don't we go for the particles that are considered compatible with us, with our animals or a living body. So carbon is the best one, and from there we started and think of, we have consider 1 year consider review and going through experimentation like that. Then we came to a conclusion and synthesized carbon dot. Later it was confirmed by us also. We sent it to Germany also because we were new because we could not understand that what we have made. We were to that stage, I and Mr. Das, and both we were preparing. So it was sent to Germany for characterization because that facility was not available in India. And later we got a very astonishing result that this is a new particle and you have synthesized it. This much properties is carrying. So it's a novel idea and then that's synthesized and later on we got. So all these processes are broadly divided into four process. So how synthesize it? Because we have taken onion, pyaj, what we are using in the household work. So it can be synthesized from palak, loki, whatever it is, a bottle gourd and palak, spinach. Can say any even garbage also kitchen waste. So any organic compound from where we can synthesize carbon dots. So it's a eco-friendly first and foremost thing that can say to our satisfaction. So we have uh, isolated this onion genomic DNA. Means we have isolated DNA from onion cells. Okay, and thereafter treated with chloroglucinol. Okay, and plus DL dopa, and it automatically converted into carbon dots which having some functional group. This right one. It takes almost 8 to 9 hours. Just we put it in autoclave because this much temperature could only be yeah. And we were continuously running the autoclave for 9 hours, 10 hours like that. Continuously autoclave is running. So it is a little bit dedicated work because to that much heating process, we call it as hydrothermal pyrolysis. It could be synthesized. So can we imagine that how difficult or how precise process it is? So we have to heat it through to that end. So ultimately carbon dot is synthesized having aldehyde group, carboxyl group, hydroxyl group, amino like that. So we can add any type of group. And here is the mystery that if we wish to target any drug, so we can conjugate with this functional group with those drugs and it will ultimately carrying to the site. Okay, here is the mystery. The later on we understood when the analysis came to us and the chemical structure and all that. So we can use it. Okay. So this is the very simple thing, not very complex that we can't understand. So this much can say functionality I did, and finally we uh, synthesize the carbon dots. Okay, so whether it is carbon dot, it is working, it is of value or not, we can't say. Just it is. It looks like a gel. So we can't say with guarantee unless and until it goes through quality check for any manufactured fabricated products, whatever we are using, it's a common phenomena that it has to be quality checked that what is inside, what it contains, what is its shelf life like that. So we also have to provide the evidence, chemical characteristic. That is the most difficult and critical thing. Synthesis of carbon, any nanomaterial is not a difficult process. In simple lab, we can synthesize it, but whether the product has fabricated in proper way, its chemical structure, whether it is useful for our purpose or not. So we have to characterize it. These are the basic rules. So once it characterized, then only it is acceptable. Otherwise, it is of no use. We can't say with guarantee that we have synthesized it all that. So for that, we have gone through several processes. Okay, here some advanced laboratory instruments or laboratories required. So FTIR we used for Fourier transformation spectrum analysis we conducted just to get that whether the functional groups are sufficient on the surface of a carbon dot or not. So we confirmed it that there is a sufficient number of aldehyde and carbonyl and hydroxyl, whatever it is required. Okay, FTIR. Then we can say analyze these carbon dots through a scanning electron microscope. Okay, so these particles are looking under the electron microscope. Okay, so it has become particulate. It might have been initially in the gross. It seems that it is a gel and nothing is particle in it. It's simply I have some graphs I'll be showing you. So it appears that it is nothing, no particle is there because it's simply gel-like, jelly-like a structure. So whether it is containing any particle or not, if these particles are not formed or it is not in a proper way, so we can't say that it is of carrying any functional group or like that. So we can say studied these phenotypic characters of the carbon dots synthesis. After that, photo physical property of the CDs were determined. All these graphs are taken steady state, excited, spectra, lifetime, UV visible. These are all criteria that are used for characterization of this nanomaterials as a whole. Okay, so we have also used that. And the most thing is important thing is that we have to use just 100-watt bulb, yellow bulb, what we used to use in our early student life. Still, it is used in some concept areas also. So it is a simple bulb जो पीला रंग का होता था फिलिप्स का वो we use that and through excitation it is a phosphorescence fluorescence property. So it emits lights of different wavelength, and that wavelength generates ROS. I am telling you. So this is all the story what we have done it for the taking the onion, प्याज से निकाला, पकड़ डाला, ये कर दिया hydrothermal pyrolysis, then DNA dots were synthesized. We hybridized it with different functional group polymer dots, and there was a story of tannic acid. I will be telling you in the later on. So here the carbon dots is prepared, okay, but it was not having some can say sticky property, and we were thinking of making it as a good bioadhesive just to replace suture what we are using in surgery. Okay, so it was not showing a sticky property. So one time, uh, I, I thought that we got failed, and we should wind up this research work. There is no way. I think we were almost hopeless in the midway of our research. After 6 months of a struggle, we were hopeless. So we thought that if we have failed, so why should worry? Once we try that, some alternates will be there. And we are studying, especially myself, all the coming from the college and sitting at the night and running through this on internet matters. And I have brought so many can say chemistry books from my friends, and I used to go to the library also just to know the chemistry of some of the organic molecules that are biocompatible. And we were totally hopeless. Frankly, I am telling you that we have failed, and there is no of can say use of working on this area. So we were just let's wind up and we accept that we have failed. No problem. So we are in that stage because nobody was to guide us. All people were having can say experience in nanoparticles, and we accidentally can say entered in this area without any guidance like that, just reading books and whatever it is, and hypothetically we have created that is a you can say whatever it is. So lastly, I collected some material that tannic acid is having many OH group. Tannic acid is having chemical structure have gone through, and the second was this vitamin E, tocopherol acetate. Both are having sticky property and both carries the functional group, OH group. So that could be compatible with the biological material. So I have tried both this I procured this tocopherol acetate, tocopherol acetate from high media, and we also procured tannic acid and mixed this with carbon nanomaterials what we have prepared. And after that, we got hope. So by adjusting the different concentration of tannic acid, and can say we left it because that was tannic acid was much more better, and we left the tocopherol and vitamin E. That it is not tannic acid could be missing. So by doing so, finally we synthesize this can say carbon nanodots bioadhesive just jelly. This one is taken from my lab. From just one sample was taken here, and I have taken photograph with my mobile. Okay, so this is how we came to the journey. Okay, so these are all about the this characterization and all that. See, this is a by opening the lid of the this vial, and I have taken photographs. See, look just like jelly we are using on the bread or jelly we are using for in our food items. It appears somewhat like jelly, kisan jelly like that. The color is a little bit different, and by touching it, we can't say that it is a particulate in nature. But through electron microscopy, we have to confirm it. Its particle size it could have been considered clumsy. So most of the parameters I haven't added here because that is a little bit complex one, and we need some basic can say idea of chemistry and all that. So I haven't included those slides. So we have also measured this carbon nanoparticle slide. So it was around 9. In one case, it was around 8.63, and second was in 9.12 nanometer. We have measured it because that is also part of this, that we have to confirm that if we are telling carbon dots, then we must be proving through experimentation that it is below 10 nanometer. So we have measured also the atomic force microscopy, and so many instruments we have been using because characterization was a little bit challenging also. And we cross-checked with the reports from Germany, what we have sent and confirmed it. Okay, so there are photophysics of we have again studied the photophysics. One was stage was synthesis of carbon dots. Second was synthesis of bioadhesive. Okay, by doing so, these are all chemical can say analysis of the yeah, and this tag test. Uh, uh, just we are using just a sticky taking concept pinch of carbon bioadhesive, and by doing so regular movement of finger, it become fibrous here. It become fibrous, and this fibrous nature actually binds the cell. Off the record, we have tested that it can be replaced with suture, but formally first actually we have to go through this lab animal testing, then we can say switch in the domestic animal and finally in the human beings. So animal experimentation is still awaiting because our lab animal has been demolished due to construction of a new building. So we are waiting, or we will be trying somewhere else in the neighboring institute like IGMS and all that. So once we prove it, but off the record during operation or some wounded animals, we have tried it. It is working without suture. It can be used for binding, but just like it is a bio glue. Means you are simply sticking it, so no need of suture, and it is providing sensation that the wound has been healed, and wound becomes self without any ointment and all that we have cured. But there was no post-operative care, no gel, no can say external ointment has been used. This is sterilizing. This bioadhesive is having self-sterilizing property and self-adhesing property. We have so probe tag test was also conducted because for that we use this probe tag test, and by simply putting this gel and stick this lever, and it is pulled, and how much force is required, it force distance curve is made on the electronic panel, and only when it breaks means strength is tested through probe tag test. It's an international recommendation for recommendation any adhesive. So we have to go through also the for this test, thermopyle examination and probe tag test, that we used, okay, for that time. And after that we tested these biodesives to see in the right panel, the skin of the pig, just we are just like we are when we are using this glue to paste two piece of paper or something like that. So we have used it just to stick it, the two pieces of pig skin collected from the field condition, and we applied here, and it was totally pasted, and thereafter we tested its strength also by pulling it through a lap shear test, because when it this breaks, this adhesion point breaks. So here left is the tool we have started it, and finally it ruptured, means detached from one another. So this force is was consulted in the graph, so that we can say that this much strength this is having. So we it can be used for bioadhesive or not. So these were totally application of this. Here we dipped it in the overnight. This pasted pig skin. If it is water resistant or not. So it is water adhesibility stability test also showed that it is working. So this much parameter we have to do it and do it. Then only science. In science, anybody hears you, believes you, once you prove it. Otherwise, just you want to tell me some. So foolproof, with foolproof, I can say determination, we have to face the science. Okay, so after gel were applied, okay, here no lights were given. Okay, in our cases, 1 hour exposure of this carbon nanomaterial, or you can say yeah, carbon nano that carbon dot based bioadhesive, it requires an excitation process that is provided through visible light. So what we are doing that can say lighting this 100-watt bulb, and in the I have used, frankly speaking, I have used egg candler. Just in biology lab we used to candle the eggs, so we use that bulb, 100-watt bulb, 200-watt bulb. So I used by fitting bulb inside it and in a candler, and I have put these plates, these my Kanke agar plates and all that in those boxes, and left it for 1 hour. Okay, and after that, what we observed, that with exposure of light, see there is no colony, means nanoparticle has concept totally inhibited the beta cells. Okay, here there was no light exposure, so some colonies were inhibited, but it was not completely concept sterilized process. And these two are control, means we have already inoculated the E. coli colony, so it was having, and the same replica without light showed little bit colonies 'cause although it has inhibited the bacterial growth, but not complete. But here, after exposure of light, 1 hour exposure to the visible spectrum causes the complete sterilization. This is a proven one. Likewise, for the Staphylococcus also, similar experience we have. These plates were taken, photographs were taken after experimentation. So I have done it by myself. So I can say that finally that it is working. Okay. So still we are working. There are so many photographs also. We have also correctly still continuing, see how much we can succeed. So by doing so, fabrication of bioadhesive, we prepared CD gelatin tannic acid conjugate. Okay. The two organism we have tested all. We are in a process of testing of some other concept like Pseudomonas and Klebsiella like that. So initially we have tested two established bacteria because they are easy to work with. So Staphylococcus aureus and E. coli. So they have shown can say inhibition against the carbon nanoparticles and the self-healing property, built-in self-healing property means simply you apply one thing will be that it will be there will be no bacterial infection because it is generating a reactive oxygen species. And second thing is that it converts into fibrous material later on, so it will be providing strength. So the ends of the wound that will be can say pulled like just a rope or a suture. So it is just total if it we are succeeding, still it is in a process. It is not easy just to say in just one experiment that yeah we are ready and you can use it. We have to go through so many processes for validation and all that. So that is a long journey, but we have at least started this journey of carbon nanomaterial. In the process, I could not have somehow I forgotten. I wish to show you the drug delivery, how drug delivery. We have diverted this after this to the drug delivery system. We have used this carbon for drug delivery also, and there are so many ways. We are also planning to try for the diagnosis and all that. So see how far we can succeed. It depends upon our work and okay, how much we can succeeding. So this is all about the work. I have shared it and the nanotechnology that I try my best just to share you my experience. Rather, initially I was thinking that I must be a servant background, but I thought that people from our science fraternity is sitting with us. So at least to motivate them. If anybody is interested in such type of research, if anybody is having some access like that, so I am ready to help. I'll be happy to can say help if anybody is initiating such processes. So I have learned by can say somehow dhakke khake seekhna, but I'll be helping because I can remember the pain we have took and so many times disheartened that we have failed like that. So this is how can say I have started. Now we have got confidence. We are working on some more projects. So okay, this is all about my work and the nanotechnology in brief. Just I wanted. So is there any query? Please, you may feel free to ask me. Hello.
Sir, thank you so much for such insightful research work you have done and you have presented here very well. And I can see in the chat box the participants are very much impressed. Many of them are asking for the PPT because they are so much impressed with your research work and they have sent commendable messages to you in the chat box. You can also see that people are
Yeah,
liking
yeah,
your work and they have enjoyed your session so much. And we have some questions in chat box also. So there is a question from Dr. Saksham, and the question is: Are nanoparticles effective against multi-drug resistance bacteria causing pyoderma in dogs?
see, from the findings, what I have done is Staphylococcus aureus and E. coli. They are totally effective without any antibiotic. We have used pure carbon dot material, no trace of antibiotics in it, and it's successfully inhibited. And repeatedly we are getting it. So, but one thing is that I can't say can say with confidence that in pyoderma, if we are using intra can say uterine therapy, so that nanoparticle will be working in those can say environment, whether the pH is different or the molecules are different. So that is a matter of can say experimentation. I haven't used it. I have only tested on the pure bacterial colonies, what we have isolated and characterized earlier. So on therapeutic purpose, it can be useful. It's a matter of trial only. So we can use it.
Okay, sir. One another question we have from Dr. Santosh, and the question is, what are the nanomaterials which are available in market for practical use? It will be very good if you mention few with their uses.
Yeah, gold nanoparticle is very much established. Gold nanoparticle most famous, most more established, and some of them are can say in use in some of the drugs like that. But you can say these nanoparticles material is also a matter of I can say IBSC and so many government norms and rules and regulation. It has to pass through it. So I think hardly one or two is can say available. That is nano gel for dressing material. I have what I know. Nano gels are available. Nano bandages like material which are containing nanoparticles of this silver or gold derived nanoparticles. So it is in use. Okay, but for oral and body, there is a can say, rigorous rules and we have to follow it. So it will require. But for topical use, it is available. Nanogels, nanogel
Yes.
based, can say, dressing materials.
Sir,
Okay.
we have another question. Somebody has asked that is this technology cost effective, and how we can use it in daily clinical practices
Yeah.
instead of such as limitation and pros over such as.
No, no, no. We haven't just we haven't considered delve into the economics because we are thinking that first we made it, then we calculated how much it cost. So two things I have also can say a question in my mind that whether it is cost effective and whether it is viable. I am at the stage of just experimentation that I have synthesized it, prepared it, and it is showing some results, encouraging results. That is a good thing. It looks better in science, but for to become a public good, this whatever material I have synthesized or worked on, so to make it a public good, we have to go through several processes, and it should be viable to the public one. Because common man is also using, and he is getting the same result what I am. So it's a long journey. We have to go through repeated process, and we have to prove it. It's validation. So till now, till that time, and self life also, because it is a very short shelf life. Because I haven't seen keeping in the fridge after 2 months it is any having any property. So within 2 months it gets loses its property. Okay, many a times I have kept it, and after 2 months like that, I that we should test it. So it's self life. So we have to determine the self life whether it is working, because just synthesizing and using within 1 to 3 days, it will be not of value, because how much we are synthesizing and repeatedly you can say supplying to anybody. So it's a little bit a question for the future also. Okay. Next, madam.
Yes, sir. Next, we have question for how much time this glue provide adhesion. It is advisable to apply it in wet area or in
Li-
dry.
Yeah, yeah. No, we can use it in wet or dry both, because it is hydrophobic in nature. Okay, so there is no can say question. we have simply applied it on the, can say, incision, at the site of incision, and left it. So it is not like that morning, evening, we are applying it just like therapeutic material. So for first time applied, and it is forever. That is should be the question, because repeatedly applying and like that, so it will know. So for one time initial stage, it is applied and left it, and after that the wound healed. What we have experienced, but we have to go through this process. Just informally we tried it, that whether it is working or not. For some cases where already we know that this is considered damaged case, and we are not damaging anymore. So why don't we try it? And it shows encouraging results. Okay.
Yes, sir. One
Next.
last question I'm taking from the audience, and the question is: Are nanoparticles safe to use in dogs? And if yes, then up to what maximum limit it can be used?
Dose has to be decided. Just now I told you, madam, but we have synthesized it, we have characterized it, we have used it, can say some animal. So after animal testing, dose, how much it should be applied, but it is non-harmful, non-allergic. It is causing even to our skin. I used to just taking some and testing by my thumb impression method, what I have mentioned here, simply doing it, and for 1 hour, 2 hours, like that, I have left it consume on my finger, and I have forgotten to wash my hands, and this has happened so many times. But 2 hours, but I haven't experienced any kind of damage or something irritating like that, allergic reaction, or any type of can say adverse effect on my skin also. On the animals which what we have applied it, because that was animal we accidentally can say the accidental case means it was lying on the roadside due to accident. Some its skin was, can say, erased, incised. Okay, incised, and due to aberration, and we thought that why we should help it, because that was poor street dog not having. So in the meantime, one of my colleague at the surgery department, I told him that this is already in, can say, such condition, so why don't we try it? If we are saving it, so that dog will be concerned not in a position to stay with us for so long. So we have tied it with in our can say OPD ward, and like that, and applied it, and for 2, 3, 4 times, that see it is happening. So it's still, I can see those, can say, animals, they are running, roaming here and there, and I secretly watch that, what is happening to that spot, and by going close to those animals, I haven't seen there is no trace, neither, can say suppose we can say there is a baldness or deherring of that area, like that. So it is not a case, but for the scientific point of view, we have to test it for the toxicity. So we will be testing. Toxicity testing is our agenda. But once we switch to the laboratory animal trial, and after that we will do histopathology and all that. So there are so many parameters still we have to work. So we are waiting for the animal houses to that will be finished, or we are getting somewhere the animals facility in the neighboring can say institution. So after that, I can finally say that it is toxic or non-toxic based on our experiment and scientific basis. But in just mota moti, common man language, I have told you, I have honestly shared the story that there is no reverse. Okay. Next, any questions?
Yes, sir. We are getting so much question, but you have answered most of them, and most of them you have answered in the presentation. So we are skipping them, and it was indeed a very fantastic session. Everybody is so impressed with the research work you have done, and I'm seeing that in the chat box. So thank you so much, sir, for this insightful session and for showcasing your research with here and sharing your knowledge with all of us. And we are very thankful to you that you have spared your valuable time and interacted with our participants. Thank you so much, sir.
Thank you,
Okay,
sir.
thank you. Thank you. Okay.
Dear participants, as we have come to the end of tonight's program, I would like to take this opportunity to thank all the valued and respected dignitaries, including chief guest, guest of honors, and our amazing speaker, Dr. Manoj Kumar. So the sharp intellect and vast experience of our speakers boosted the knowledge of our participants, and I think indeed it has happened. So we feel very privileged and that we have listened to all of you and learned from you. And in case of any technical glitch which has happened, I apologize to all of you. So, dear participants, thank you for joining. Thank you for being attentive and asking the questions and making this program interactive. So, we will join tomorrow, and the details will be shared in your respective WhatsApp group. So, thank you, everybody. Thank you for joining and making this program success. Thank you all, and have a great evening.
Thank you. Dr. Dinesh, one request is from my side. Hello, Dr. Dinesh. Hello.
Hello.
Dr. Dinesh, are you getting voice?
Hello.
Thank
you, sir,
Dr.
for
Dinesh.
your nice presentation.
No, just I'm thinking that if anybody is having curiosity or he wishes to work on it like that, some of the faculties are like that. So collect those feedbacks or whatever some want some more information from my part. So get their feedback and communicate it to me. Okay, I will be trying my best. Okay, thank you.
Thank you, sir. Thank you once again. Thank you, everybody.